So let us discuss atomic absorption spectrometry. It is an analytical technique that measures the concentration of the elements in the given compound. Atomic absorption is so sensitive that it can measure down to the parts per billion of a gram in a sample. So we can say the sensitivity is high for this method. Then the technique makes use of wavelength of light specifically absorbed by an element, and that is why the name is given, that is a absorption spectrometry. Okay, so it makes use of wavelength of light specifically absorbed by a particular element, and they correspond to the energy needed to promote electrons from one energy level to another higher energy level. Now it corresponds to the energy because the electrons need energy to go from the lower energy state to the higher energy state, and this is with the help of some we can say trigger. Okay, so it is related to the energies needed to promote electrons from the one energy level to the higher energy level. Then the atoms of different elements absorb characteristic wavelength of the light. Now the atoms of different element, for example zinc or copper, iron, each atom have uh, actually those elements have different atoms, and different atoms of their elements have characteristic wavelength. So that wavelength can be absorbed, and that is used in this technique that is applied, and the principle of this technique is depending upon that, and analyzing a sample to see if it is contain a particular means using light from that element. Okay, so if it contain a particular element, okay, analyzing a sample, that means whether it is having ability or whether a particular element is present in the compound. Okay, now in atomic absorption spectrometry, the sample is atomized. That means atomized means it is an element and small atoms are present. So the sample is atomized, that means converted into the ground state, free atoms in the vapor state. Okay, so the atoms are converted into the free atoms, and that is in the vapor state. Okay, initially it is available in the sample, but it is a atomized, and the beam of electromagnetic radiation emitted from the excited lead atom is passed through the vaporized sample. Some of the radiation is absorbed by the lead atoms in the sample. Okay, now this is example for the lead, okay, as a element. Okay, so some of the radiation is absorbed by the lead atoms in the samples. Okay, and the greater number of atoms there is in the vapor, the more radiation is absorbed. Okay, so if the number of the atoms are high, great, then the more radiation will be absorbed. Okay, the amount of light absorbed is proportional to the number of lead atoms. Okay, then the amount of standard absorbed is compared with the calibration curve, and this enables the calculation of the lead concentration in the unknown sample. Okay, so at the end we have to calculate the calibration curve, and from that we will able to calculate the this concentration of the substance. Okay, the common light source is hollow cathode lamp. Now in this you will see the ionization method is there, the sputtering method is there, excitation and emission. Okay, so all these methods are there. Ionization is there, then sputtering, that means attraction towards the particular electron which is there in the light source like the cathode lamp. Uh, in this atomic absorption spectroscopy, hollow cathode lamp is used. So in sputtering, the atoms or ionized things will get move towards particular things like M0 or Ne plus. So this is a sputtering, then excitation of the electrons, and then emission. Okay, and the light selected by the monochromator is directed onto a detector that is typically a photomultiplier tube. Okay, and this is a grammatic diagrammatic representation or instrumentation of the atomic absorption spectroscopy. So hollow cathode lamp is there, then atomizer flame is there. Now the sample solution, it is given there, the sample solution, nebulizer, and the flame. Okay, so sample solution will go into the nebulization. There is a formation of the small particles, and when it is bombarded in the flame, it will give a certain atoms, and then when they fall on the monochromator, it will detect a particular absorbance. Okay, so that absorbance is detected and the recorded. So this is all about the atomic absorption spectroscopy. Okay, now the applications like the clinical analysis, atomic absorption spectrometry is applicable in clinical analysis, then environmental analysis, then pharmaceuticals, and mining. Apart from that, various industries like construction industry, atomic absorption spectroscopy is used. Like for example, clinical analyzing metals present in the biological fluids such as blood and urine. Okay. Then environmental analysis like monitoring our environment, finding out levels of various elements in rivers, sea waters, drinking water, air, petrol, drinks, wines, beers, and fruit drinks, etc. Okay. Pharmaceutical, we know for the analysis purpose in some pharmaceutical manufacturing process, minute quantities of a catalyst used in the process are sometimes present in the final product, and by using this method, the amount of catalyst present can be determined. Okay, so all these are considered as an application. It is widely used for the elemental determination. Okay, so with this, we will end our today's session.